My name is Arya Sadafi and I'm coming from Helmholtz Center in Munich. I'm also affiliated with Chair of Computer-Aided Medical Procedures at TUM. Today I'm presenting our work on sickle cell disease severity prediction from Percoll gradient images using graph convolutional networks. Sickle cell disease is a good example of natural selection process. People with one mutated gene are less likely to die from malaria, while people with two mutated genes are experiencing complications and anemia. In Africa, up to 40% of people are believed to carry one of the sickle genes. Severity monitoring and prediction of the sickle cell disease is an important task with currently no established therapeutic strategy. Changes in severity allow monitoring of the treatment efficiently. This assessment of severity is very important as it directly relates to the probability of complications in the future, making it a vital checkup for patients, giving them heads up for possible need for medical care in short future. Blood samples are easy to obtain and Percoll is a cheap and accessible solution. Severity grading of sickle cell patients is a challenging and time-consuming task, requiring expertise and equipment that may not be always available in affected areas. Using the Percoll solution, patient's blood sample and a centrifuge Percoll gradients are obtained. Several bands are formed on the tubes and cells are separated based on their density. In addition to Percoll gradient images, we are provided with some clinical data consisting of the spleen size and percentages of hypo and hyperchromic cells. Hypochromic cells are having a lower density of hemoglobin, while hyperchromic cells have a higher density. These two values are important in sickle cell disease because having a lower density of hemoglobin means less chance of sickling and thus less severity. A function in the spleen helps clearing of the bad cells from circulation and less chance of complications for the patient. The dataset has 216 samples consisting of the Percoll gradient and the mentioned clinical data classified in 4 different severity grades by the experts in the hospital. Since we are aiming for an affordable and versatile solution, we decided to estimate the hemoglobin density directly from the Percoll images and avoid depending on expensive lab equipment. For this purpose, a convolutional neural network is used for regression on those two values. We used mean square error loss and achieved the RMSE of 6.5 for percentage of hypochromic cells and 0.9 for hyperchromic cells. So now our approach to estimate the severity of the disease only depends on the Percoll image and the size of the spleen obtained and reported through an ultrasound examination. In our previous work, we classified Percoll gradient images into 4 classes of sickle cell disease: control, thalassemia, and hereditary spherocytosis, demonstrating the capacity of the Percoll gradient method and the information it contains. We use the same trained AlexNet model working with the fusion of the features obtained through the channel-wise fast Fourier transform to extract the features from Percoll gradient images. Having the features extracted through the mentioned method using the hemoglobin density estimator, we estimate the percentages of hypo and hyperchromic cells to formulate a similarity metric based on dose and the spleen size between every sample in the dataset. Based on the similarity metric, a population graph is constructed. After some graph convolutional layers, the severity score for every individual in the graph is estimated. We used 10-fold cross-validation and performed 5 independent runs. Accuracy, F1 score, and area under ROC is reported. We are using SVM as a baseline, processing only feature vectors. Same SVM having feature vectors and additional clinical data. The GCN with random clinical data. Our proposed approach with estimation of hypo and hyper percentages from the Percoll images, and finally the GCN with ground truth hypo and hyper values measured in a laboratory which is outperforming the previous approaches as expected. Results look promising and provide a solid ground for future work. Giving up only 4% of accuracy, we can become independent from the laboratory, which is not accessible in many remote areas. We performed the second ablation study to evaluate the significance of the selected clinical data, and as expected, the combination of all 3 parameters yields the best result. Additionally, looking at the UMAP analysis of the feature vectors, our suggested method, and the GCN with clinical data measured by the lab, we can see that both of the Proposed GCN-based approaches are able to separate the samples according to their severity, and in our proposed approach, a clear spectrum of severity is formed. However, this approach has limitations too. The limited number of patients available makes our cohort relatively small, and experts have contradictory opinions about the severity of every case. More experiments are also needed to ensure the robustness of the model across different illuminations and cameras. Here we propose the first computational method requiring only the Percoll gradient image and a spleen size obtained from a conventional ultrasound. Analysis of Percoll gradient images with CNNs nicely predicted percentages of hypo and hyperchromic cells, and the proposed GCN predicted sickle cell disease severity score with a surprisingly high accuracy. In future, we will analyze more patients, especially more severe ones, as well as pediatric datasets. I will conclude here and thank you for your attention.